In the second part of chapter 13, we are going to look at some chromosomal disorders that can be inherited. This is not going to explain all genetic diseases, but it will be a start to explaining them. So we're going to look at a thing called a karyotype. That's really important for examining the number of chromosomes that are around. We'll talk about this process called non-disjunction. That's when things don't divide properly. And then we will look at aneuploidies. Those are abnormal numbers of chromosomes. And we'll talk about errors in chromosomal structure that can occur. So where is this happening? Well, errors in meiosis can lead to a couple of different outcomes. You could have an abnormal number of chromosomes that are resulting from errors in meiosis. That's one of the things that could occur. You could also have problems with the structure of chromosomes, bits that get swapped when they shouldn't, and that is equally as bad. To examine this, we need to have some way of visualizing the chromosomes. That's where a karyotype comes in. A karyotype is a microscopic picture of dividing cells, and the chromosomes are basically just cut out and lined up to check their length, the banding pattern on them, the centromere position, and if they have a proper number, right? So here you can see a karyotype that's using fluorescence. We can see fluorescent bits on each of them where they bind, and we can check these against a standard reference and see if there's any problems. In here, to my eyes, this is looking pretty good. There might be an issue on chromosome 21 there, but this is looking standard for number, right? So in humans, we have 22 autosomes, non-sex chromosomes, and they've been numbered based on their size. So chromosome 1 is the biggest, 2, 3, 4, so on and so forth, down to 22, which is the smallest in length. Then we have the sex chromosomes, which are separate here. So chromosome structure is important. This gets looked at a lot in the karyotype. It's like an early thing to determine if there's any issues in there. And while not as important now that we have things like PCR and stuff like that, a karyotype is still very, very important. So here's an actual image of a chromosome. You can see all the coiled chromatin in here. And the two sides are chromatids, right? And they connect up at what we call the centromere, which is this constriction here. That's where the spindle fibers attach and they get pulled apart. By convention, the shorter arm of the chromosome gets called the P arm, and the longer one gets called the Q arm. Not super important for us, but oftentimes in a 300-level genetics class, they'll ask about these things. So we said that chromosomes don't normally look like this, right? When the cell is outside of division, they're uncondensed and they are not really visible under a microscope. So how do you get a nice karyotype, right? Remember when we looked at slides in the lab, we didn't always see dividing cells. How do you guarantee you get a lot of nice dividing cells? Well, this is where a certain chemical called colchicine can come in. Colchicine can stop cells in metaphase. Okay, this is toxic to you normally, but if you take some cells and they're dividing, you can stop them in metaphase. So for many organisms, this is very, very useful. You can apply colchicine and you stop them from pulling apart, and you can look at the different chromosomes in here. Now, there's some background stuff in here, but if you look at enough cells, you can get some nice images and you can actually cut all those things out and check whether the chromosomes are there. You can also put it in a hypotonic solution, which will cause the cell to swell, separating out the chromosomes. So there's lots of various tricks to getting a very nice image from our karyotype. So in this case, we're looking at something that has 8 different types of chromosomes. I'm going to guess this is a bird because they're using Z and W, which if I remember right, is the sex chromosomes for birds. So you'll see these little background bits in here. These might not be real. They might be bits of membrane or something like that. But we have our chromosome 1s here. We have our chromosome 2s there, chromosome 3, 4 is in here somewhere, maybe. 5, 6, 7, 8, and Z and W are the sex chromosomes. All right, so that's how we actually get the cells at the point that we want. We use this chemical colchicine. But what can happen when things go wrong? Let's look at that. So let's talk about this process called non-disjunction, when homologous chromosomes or sister chromatids fail to separate during meiosis. We're going to talk about this in the context of meiosis because we care about this because that's going to get passed on to our offspring. That's what we care about most. An individual cell in mitosis having this happen, usually it will get detected and then go through apoptosis. But in meiosis, if this doesn't get detected, bad things can happen. So a non-disjunction is when homologous chromosomes or sister chromatids fail to separate during meiosis. Let's look at what happens in meiosis 1. If we have a non-disjunction, we have two different types of chromosomes here. Let's say that these two homologous chromosomes fail to separate. Well, that's going to lead to a big downstream change because these two are going to get pulled over to this side, and then in meiosis 2, they'll get pulled apart. What are we going to end up with? Well, two of our gametes are going to end up with n-1, where n is the normal number of chromosomes. In this case, it should be 2. We only have 1 in each of these. And then in the other two, we're going to have n+1. So n is our normal number of chromosomes. 2, well, we actually have 3 types of chromosomes in there. That imbalance could cause problems. A non-disjunction in meiosis 2 is a little less problematic in terms of overall production, but it still leads to negative outcomes, right? So in this case, we separated fine in meiosis 1. This side, very unlikely it would have a problem when this side has a problem. That would be very extremely rare, but here this side is divided properly. Here's n, we have 2 chromosomes in each of those gametes. Here we have a non-disjunction in meiosis 2, and we end up with an n-1 and an n+1 gamete. Either way, if this happens, and then these gametes go on to fuse with an egg or a sperm, depending on which one they are, that could lead to an abnormal number of chromosomes. So we have terms for this. Euploidy, U means true. Euploidy is the correct number of chromosomes for a species. We would then call an aneuploid and something that deviates from that number, right? So a not true ploidy. This is a term that doesn't tell us everything about it. We could more specifically describe like monosomy, missing one of the 2 chromosomes, right? Or trisomy, having 3 of one of the chromosome types. So those are a little more descriptive terms for aneuploidies. One of the most common ones that we hear about that is not lethal, because in humans, many times this will be lethal, we have trisomy 21, which can cause Down syndrome. The risk of trisomy 21 actually increases with the age of both the mother and the father. It correlates with these things here. I'm showing maternal age. You can see the percent of births that have trisomy 21 involved. At past age 40, that goes up quite dramatically. Trisomy 21, you can have a viable offspring that does survive. They do have developmental differences, but they do survive. Oftentimes, though, other aneuploidies and other chromosomes lead to gene dosage issues, which do not result in a viable offspring. Trisomy 21 can be detected by the extra chromosome on here, and this is detected in Down syndrome. But there is another way Down syndrome can occur. We can have what's called a translocation, where actually part of chromosome 21 breaks off and attaches to another chromosome, in this case to chromosome 14. That effectively leads to 3 copies of chromosome 21 in the individual, and that dosage is what leads to Down syndrome. So these trisomies are common ones in humans, trisomy 21, as we saw, 0.5% roughly of births can occur for older mothers and older fathers as well. It's not just the mothers. This is an example of one chromosome having too many or too few, right? In this case, too many copies of chromosome 21. Well, what happens when the whole genome gets duplicated? This leads to what we call polyploidy. Polyploidy is more than 2 copies of each chromosome. Polyploidy in animals is quite rare in my experience, and often it does not result in viable offspring. Polyploidy in plants, though, super, super common. It has some really interesting things that happen when you have plant polyploids. So here we have a tetraploid. It has 1, 2, 3, 4 copies of each of the chromosomes in the genome here. So let's talk about the first example, triploids. Well, triploid is problematic because triploid has 3 copies of each chromosome. So that's problematic because when you go into meiosis, you're going to have a hard time dividing equally, right? You have 3 things. How do you divide those into 2? Well, you can't have 1.5 chromosomes in each. So it's going to lead to improperly balanced meiosis, and that often leads to sterile offspring. Basically, the cells don't divide properly. It leads to sterility. That might sound bad, and in a natural context, yes, not being able to reproduce is a negative, but it's actually kind of useful for a couple of contexts. 1, conservation, and 2, commercially. Okay, so triploid fish. Triploid fish are viable, but they will not reproduce. How is that useful? Well, you might be familiar with the practice of stocking lakes and streams with fish. If we're stocking them with non-native fish, we can stock a lake with recreational fish, but they can't reproduce, and that means they can't get out of control, and conservationally, they're not going to wreck the ecology, right? If you like seedless watermelons and seedless bananas, here's a seeded banana. That doesn't look very appetizing. Those seeds are giant. Here's a seedless banana. Seedless watermelons and seedless bananas are triploids. How do we get that to happen? Well, we can actually induce this during meiosis. We can apply chemicals or currents to them and stop that from occurring. Then, because plants can reproduce asexually, you just cut off a branch, stick it in the ground, and you keep propagating that tetraploid thing. So commercially, this banana right here is much more appealing than that one right there, right? Another interesting thing happens in plants, though. So the triploids are problematic for natural populations, but even-numbered polyploids are actually very, very common. And something strange happens. As the genome number increases, as the ploidy increases, plants will often get bigger in physical size. Why this is, we still fully don't understand it, the dosage issues and things like that, but it is a well-documented phenomenon and very commercially important. So you go out in the wild and you find a strawberry that's a diploid. It looks like this, right? It is nothing like the giant monsters that you find in the grocery store, which are octoploid, right? They have 8 copies of the genome in each cell. Here we have, I think maybe this is alfalfa or something, some sort of animal feed, roughage. We're looking at a diploid, and then the tetraploid here is much bigger, right? You can see this clover here. We're getting a much larger size in the tetraploid here. Even in Arabidopsis, the tiny little weed that we study, here's a diploid, here's a tetraploid, here's an octoploid. You can see an increase in size here. Super interesting, super important. I think I've mentioned this probably several times now, but the most I've ever seen in terms of ploidy, I saw a 32N ploidy once, where it had 32 copies of the genome in each cell. That's on the extreme end, but plants are really flexible to polyploidy. Animals, not so much. All right. In our karyotype, you probably noticed the sex chromosomes, right? 2 X's in humans leading to biological female, an X and a Y, which is this stunted little thing here, lead to a biological male. This system holds for a lot of mammals. This causes a problem, though, right? Because females are going to have 2 copies of the X, whereas males are going to only have 1. And as we've said, gene dosage can be a problem. So to keep the dosage of X's the same between males and females, an interesting process happens. This is called X inactivation. Early on in female development, in certain cells, one of the X chromosomes basically gets inactivated or shut off. It winds up and becomes heterochromatin. So which one? It's actually random. Okay, the pattern, is it the maternal one or the paternal one? It's random in different cells. So you can actually get what's called a chimeric phenotype from this. So let's look at this in cats. Here we have a cat, and this is a tortoiseshell cat. So it has orange fur and this darker black fur here. Why is that? Well, these pigmentations are linked to the X chromosome. It inherits one from the maternal lineage and one from the paternal lineage. On one of them, we have the allele for orange fur. On the other, we have the allele for black fur. During cell division, X inactivation occurs. In these cells, the black-furred X chromosome got inactivated. It gets wound up into heterochromatin and is not expressed. The orange fur allele gets expressed. But in these cells over here, the opposite happened because this is random. So the orange one got inactivated and the black one is being expressed. So it's not always the same X chromosome that gets inactivated, so different alleles might be expressed in different cells, which can lead to different phenotypes. Kind of interesting. This also happens in humans as well. There are cases where we see differing numbers of sex chromosomes. Sometimes these lead to developmental differences, physical differences. Sometimes it doesn't. One of the examples that we have here is XXY, where you have two X chromosomes and a Y chromosome. This is looking like a biological male, but they have multiple X's. And this causes what we call Klinefelter's syndrome. So there's an interesting website here, livingwithxxy.org, that has stories from people with Klinefelter syndrome. Many of these people had no idea that they had this until they were karyotyped. And through hormone treatments, they can live what they feel like is their normal male life. This leads to interesting questions about gender and sexual development that we're not going to address in this course, but it is something to think about. There are chromosomal bases here for developmental differences, obviously. So Klinefelter syndrome, as you could see, you may not even know that you have this unless you have a karyotype done. There are some other examples here, like Turner syndrome, which is also called XO syndrome, where you have a single X chromosome, no Y chromosome, or no second X chromosome, where there are some larger developmental differences, including some that can be dangerous, issues with the aorta, kidney development issues, infertility, things like this, and various other developmental differences. So duplications or deletions often lead to issues. So here we've seen a deletion of part of a chromosome, and this is Karyotype syndrome. It is characterized, this in French means cry of the cat. It's characterized by a typical developmental pattern, and babies often cry in a way that resembles a cat there. So there are nervous system differences and physical feature differences in this example here. This is not a full loss of a chromosome, it's loss of part of a chromosome that's occurred here. So you can see the small deletion there. You could also have duplications, as we saw in Down syndrome. Small portions of the chromosome being duplicated could also lead to developmental differences. So there are some terms for different rearrangements of the chromosomes. Occasionally chromosomes will have inversions, as we call them. Part of the chromosome gets flipped. This could be the result of like mechanical breakage or these things called transposons, jumping genes. So somehow the chromosome breaks and doesn't get fixed properly. A pericentric inversion includes the centromere. So here you can see the normal patterning, and here we've flipped around the centromere. A paracentric occurs outside of the centromere. So here you can see a paracentric inversion. Not super important for us, but these terms do get thrown around sometimes. So it's good to know what's happening and to know the term centromere, right? Because that's a key structural component of the chromosome. So interestingly, sometimes inversions don't prevent meiosis from occurring. They just make it a little bit more difficult. So here you can see there has been an inversion here in this blue chromosome, and it's pairing up normally with the other normal version of the chromosome, but it's made this loop that occurs. So these were documented. Oftentimes, Barbara McClintock in her corn lines would see things like this where there would be inversions, and she would still see that there could be recombination or pairing of chromosomes and things like that. So this does happen. It doesn't prevent meiosis, and it might lead to new combinations of alleles and things. So there is some evidence that inversions might be drivers of evolution over a long-term scale. If we look at the human genome, human chromosome 18, and chimpanzee chromosome 17, we actually see similar genes in there. And when we look at the banding pattern, it looks like perhaps there was an inversion at some point. And if you look at orangutan and gorilla, there are the same structure as chimpanzee, but humans have had this inversion. Could this be part of what makes us different from the other great apes? Possibly, totally possible. Could it just be a quirk of evolution, right? It happened and we survived and other things led to our evolution? Also equally possible. And that's one of the big questions in evolution, right? Is like, what led to these changes? What leads to the differences? And we're still working that out, even though we've sequenced many, many genomes. So a translocation is our final thing we're going to talk about here. A translocation is when a piece of a chromosome breaks off and moves. This could be reciprocal, this could not be reciprocal. So in a reciprocal translocation, one piece breaks off and another piece breaks off and they swap places. That's a reciprocal translocation. A non-reciprocal would be like this part of the blue breaks off and it tacks on to the end of the orange one here. As we saw, that can occur in the cause of Down syndrome, right? So it can effectively make 3 chromosome 21s because the genes that are in there get extra copies in there. So translocations, also another way that bits can get swapped there. A reciprocal translocation, though, is possibly a little bit less damaging because you still have all the information in there. There's no loss of genetic information, although position of genetic information does matter quite a bit. Okay, so in this modern age of sequencing DNA and sequencing genomes, why do I talk so much about chromosomes? It's still an easy way to detect certain kinds of defects, right? You can do a karyotype very, very easily, and you can see if there are changes in the chromosomal number. That could indicate that there might be a developmental difference that's going to occur. So you can do this prenatally. You can do amniocentesis, where you sample some of the amniotic fluid and look for cells for the developing offspring and see if they're going to have a normal or abnormal number of chromosomes, and then plan accordingly for what you want to do with that. It is important to understand that this was all we had for a long time. Until we could sequence genomes, we couldn't, in a large-scale way, look for smaller things. So looking at chromosomes is not going to catch everything. If a mutation of just a single base is enough to cause a disease, it might not show up on a karyotype. But this was our first step into linking heritable genetic disorders to our genome. Let's finish up with a review question. So an error in meiosis where chromosomes fail to separate is known as what? Go and pause the video, think about that. Okay, when they fail to separate, we are going to get a non-disjunction. We call that a non-disjunction when they fail to separate. Okay, so we're worried about errors in meiosis. They can lead to abnormal chromosome numbers or chromosomal structural rearrangements. A karyotype is an image that lines up all the chromosomes to check for abnormalities in there. Aneuploidy is the term we use for a deviation from the correct number of chromosomes. Remember, we said that was euploidy, so aneuploidy is not euploidy, basically. A polyploid has more than 2 copies of each chromosome. It's not just one chromosome that has extra, it's all of them. So we saw triploid and tetraploid. X inactivation, this happens in, I'm going to say mammal females, where one of the X chromosomes gets shut down randomly to keep the balance equal. We talked about inversions, flipping of the information on parts of chromosomes. We talked about translocations, where part of one chromosome moves to another. All right, that's it for chapter 13.